Ben Pascoe. I work at the University of Bath and I'm a research fellow there. And I also head up the sequencing center. And I was going to talk a little bit about a paper we've had accepted during the lockdown on our Campylobacter work in Peru. So in order to relate it all to chicken, I needed to include this slide. So I think following Fran and Martin's talk a couple of weeks ago, we're quite aware of how important Campylobacter is in poultry infections. And in the UK, if we go to the supermarket, a lot of the chicken meat we buy will have Campylobacter on it. And Campylobacter is probably, well, Campylobacter infections in people, I think Fran had it up to 80% 2 weeks ago coming from chicken. I don't know if it's quite that high, but that seems good today. And usually in the UK, it would be infections in young people or old people. And we see these seasonal differences. So in the summer, when we're all barbecuing, we see these peaks in infection. I think in the chickens as well, we see more carriage in the summer. So a lot of human Campylobacteriosis will come from chickens. And in the UK, it's predominantly a food poisoning bug. But today, I'm going to try and think about it not as a food poisoning bug. So that most of the work that we've done on Campy will have been done in the UK and America. And when we look on PubMST, which has lots of MLST profiles and whole genome sequences, more than 3/4 of those deposits will be from the UK or Europe and American studies. What is happening in the rest of the world? Is it the same in the rest of the world? As we think, or if you look back through the literature, you see evidence that this epidemiology is different when we leave developed countries. In this old Mexican study, they were looking at antibodies for Campylobacter in young children. And a lot of these young kids would have been exposed to Campy at a young age, maybe not showing symptoms, maybe not, not from food poisoning, from dirty water and hygiene conditions. So the second in this, these are some of our holiday snaps from the Gambia. And when we went, it was in the middle of the flood season. So you can see that there's flooding in the villages. This is one of the toilets. This is the toilet. This is the drinking water. And you don't need a lot of imagination to realize that when everything floods, they're obviously going to come together and cause lots of infections. In the compounds, they keep chickens and they just run around in the compound. There's no chicken coops. The kids are playing with their chickens, and there's a much closer interaction between chickens and humans. So we could see that as a possible transmission route. But actually probably a lot less through eating contaminated meat products like we get in the UK. Because they'll be eating less meat, they're not necessarily buying their chicken from the supermarket, buying a breast of chicken that's got Campy on it and undercooking it. They're killing their own chickens in the backyard and cooking things that way. So the transmission routes are different around the world, and we see these differences in the epidemiology. We see the asymptomatic carriage and chronic infection. So yeah, and this is what we've done now as part of our group, is to try and go out there and look around the world, outside of Europe, outside of America. How is Campylobacter behaving in these countries? What are the transmission routes? Do we see this reflected in the genomes? So we see different unique lineages when we go to different countries. We see the absence of other lineages which we might expect to see. In countries where you can self-medicate with your own antibiotics, we see a lot more antibiotic carriage. In the UK, we don't see a lot of multi-drug resistant Campylobacter. And then also with these detailed studies, we can start to identify lineages that cause more severe infections or even asymptomatic infections. Is there human-to-human transmission? And start to look at these other questions that are not necessarily based around foodborne, eating your undercooked chicken at a barbecue, getting ill. And that's what we're going to focus on today, is our work in Peru with Margaret Kosek's group. So she's based at the University of Virginia and also in Quitos in the northern part of Peru. And I think Francesca was on the call 1 week as well, and she was also part of this work. So it all started, 7 years ago, when we read this paper from Margaret Kosek's group, where they have this study where they're collecting samples and Campylobacter, E. coli, Shigella on a routine basis from children near the Amazon, from these sort of semi-rural settings, and just compare with detailed medical information as well. And that what they found was that they were often sampling Campylobacter, but that the children were showing no signs of any symptoms. They didn't have diarrhea, they didn't have any pains, that sort of thing. And that was interesting for us because that was an unusual epidemiology, something that we hadn't really thought much about in Campylobacter. Are we able to carry Campylobacter asymptomatically? So I think it was Guillaume, emailed Margaret and asked if we could have access to the samples and work together. And she sent them to us, and we sequenced all the genomes. And on this tree are all about Peruvian isolates, the red and green dots. And then there's a bit of context as well with the grey dots. So they're just from other studies to give us the genetic picture of Campylobacter jejuni. So we've got everything covered. And it's a typical Campylobacter jejuni tree with these two large grey blobs and our host generalist lineages. These are common all around the world in chickens and human infections, cows, and in the environment. This section of the tree we usually find associated with chicken isolates, lots of chicken isolates. They also do cause human disease. And then in this part of the tree is a bit more unknown. And you can see that there's not many grey dots in this part of the tree, but we do have a lot of our Peruvian isolates in this part there. And that's what is reflected in this plot here. So the red section at the bottom, so we've just ranked our isolates that we found in Peru and their ST by how common we found them in Peru and how often we find them in these large PubMLST databases and others. So if you're in this part of the plot, these are common all around the world. And this is where we find some of the STs that we've read about, ST45, ST21. And in this part of the plot, these are less common around the world, but we're finding that they were very common in our Peru dataset. And this is often what we see with other species as well. So we see globally dominant clones of Staph aureus that we find everywhere in the world, or we see specific clones that are dominant in certain diseases with E. coli, X-PEG. And that Campylobacter is just the same. And we see these common global lineages and then these less common regional different ST types. All your faces in the way there. And this is what we're seeing in this as well. Is that when we look in a big, massive collection of Campylobacter genomes, some of them, a small number of them we find all around the world on 6 continents, and then other isolates we just see in the particular country or the particular continent. And as we mentioned earlier, this is going to be affected a lot by sampling because we've only really sampled well from the UK and America and other countries with their surveillance systems sequencing genomes. And then there's a little bit of a diversion and back into chickens. So 2 of these common globally disseminated lineages are ST21, ST45, and they're host generalist lineages. They're common in farmed chickens, they're common in human disease, they're common wherever we perform our Campylobacter studies. A few years ago, we wrote this paper where we looked at Campylobacter from different parts of the poultry processing system, and you could see that some STs were common in chickens but less common in disease, or not common in the chicken but then caused a lot of disease. But these 2, ST21 and ST45, were common throughout, maybe less so it's on chickens than ST45. And so much so that when we looked in another study at Campylobacter from seals, it was a massive red flag to find these lineages in the seal isolates. So I think it's the pink dots on this tree. And we found pink ST45 isolates, and we were not really expecting that from seals. And when you do your source attribution work, you can attribute it back to humans and see how that we were actually infecting these seals. And when we looked a bit closer at where these seals were sampled, they were sampled downstream from where there was a big sewage, effluent discharge, so that they were getting it from us. And I think Martin and Fran showed a couple of weeks ago as well that these are the two of the most common they find when you do these surveillance studies in Oxford. When we do them anywhere in the UK or Europe, these are often two of our most common lineages. But in Peru, we didn't really see them at all. But these two, there was a few isolates, but nowhere near as many as we would usually expect to see from this number of samples. And in fact, most of the samples that we collected in Peru were from sequence types and lineages that are rare, that we hadn't seen very much of before. And that kind of makes sense and fits our narrative. We're seeing a different epidemiology, and then when we've sequenced the isolates, we're seeing different lineages that are common. And then this is a little bit more complicated way of looking at that exact same thing. So these plots are made by clustering accessory genome elements. So in this block here, we've got the Peru and the reference data set, and we see these blobs, and there's 4 or 5 blobs in this plot. But when we just look at the Peru isolates only, we don't see as many of these clusters. We don't see as many of these blobs. There's sections that are missing. There's parts of the gene pool that the Peru isolates haven't got in this population. And again, this is the same data, but just visualized a different way. And the Peru isolates all cluster in the middle, and they don't have access to this part of the gene pool. They don't have these lineages in this population. If we switch a little bit our focus to the asymptomatic isolates, so as I said, in this study in Peru, Margaret's group were collecting lots of metadata with the samples, so we were able to give them a severity score. So if any symptom that the children had, they would get a point on this score. So we'd have severe isolates that scored highly, and asymptomatic isolates with no symptoms that would get a 0. And this plot is just showing the STs according to this severity score and how common they are in these large worldwide data sets. And the asymptomatic isolates are in green, and they were all positioned down in this part of the plot. So they didn't show any symptoms, but they're also quite rare. They haven't been seen very often before. And then if we compare, we thought we might see more resistance genes or more virulence genes in the Peruvian isolates that were more symptomatic, that was more severe infections. But that really, that wasn't really the case when we compared those two together. So this is AMR genes, this is virulence genes, but they did have a lot less than the other reference isolates. So they're different from the other ones we've seen in the rest of the world, but maybe they're not so different from each other in this case. And then this is just those same things plotted on the tree. And I've made a note at the bottom here for one of Francesca's papers, because she's also looked at this same population, but having collected isolates a few years longer than we've included in this paper, and she actually sees a lot of antimicrobial resistance in this population. And if we get as far as that in the talk, I can maybe try and explain why she sees a lot of antimicrobial resistance and we don't see very much at all. So the next thing we wanted to do with this group of isolates was to try and estimate the source. So we mentioned that you can do these source attribution studies and try and predict where your human infection came from, whether it came from a chicken, whether it came from a wild bird, or whether it came from a cow. And the reason we're able to do this is because of the population structure of Campylobacter. So you see these clusters. So in this tree, white clusters are human infection isolates, yellow are chickens, blue are cows, and black are wild birds. And in some clusters, they're all the same color. So you can see that if you were this person, you could pretty confidently say, you picked it up from a wild bird. And there's a few clusters like that. So this guy's probably got it from a chicken, and this guy's probably got it from a cow. But then in these other clusters where they host generalists, we see a mixture of sources, then it's more difficult. But you can do this based on gene by gene, or even SNPs, and you can try and give a quantitative prediction of where you think your human isolate came from. And Fran showed last week that if you use different software, you can get different answers as well. But anyway, we use some of this in other studies to show that we see differences between countries. So in France, more of their infections will come from eating beef rather than eating chickens. And then previously we've shown differences between America and the UK as well. But in this case, we just wanted to try and get a simple prediction of where these Peruvian isolates were coming from. And when we performed this type of analysis, it almost overwhelmingly was predicting that all of the infection isolates came from chickens. And that made sense because when you go to Iquitos, they've got chickens there living with them. They've got chickens in the garden, and they kill their own chicken and eat the meat that way. There's not a lot of other meats available, so you can see how most of the infection will be coming from chicken. But one of the issues in this study was because of the STs that we did find, there wasn't a lot of information for us to compare with. So this type of analysis relies on what you're comparing with. Does this isolate look like other isolates from a chicken? Does this isolate look like other isolates from a cow? But in this case, we haven't seen these lineages before, so we're guessing. We're not being able to predict as well as we would like to where these isolates came from, and especially in this lower part of the tree here, this grey is basically unknown, and we're seeing a lot of grey, but we're not being able to predict where these isolates came from. So yes, in our Peruvian study, we were able to identify that there was these globally circulating lineages that we did find a few of them in the Peruvian Amazon samples, but mostly we found these rare genotypes that we hadn't seen before, which reflected this different epidemiology that we hadn't really thought about before. And then when we look at the core and accessory genome, we see that these rare lineages are not coming into contact with these global lineages, so they're not accessing the same parts of the gene pool that isolates in the UK and America will be accessing. And that a lot of these rare lineages were linked with asymptomatic carriage or chronic infection. I didn't really talk about different loss types and capsule types, which is in the paper and for you to go and read, but we also saw that some of these rare lineages had new loss types and new capsule types that we haven't seen before, which kind of makes sense. And then when we try and attribute these samples to figure out the source of infection, they were mostly attributed to poultry infections. But again, because of these rare lineages, we've got less confidence in that. It could still be that it's the water. It could be another unsampled part of the ecology in that region, another meat, another source of infection. So yeah, so what we continue to work with Margaret's group in Peru, and since then, the paper that they published 7 years ago, that's the isolates we've used in this paper, and they've continued sampling ever since then. So now there's a lot more samples for us to use, a lot more samples for us to do other types of analyses. and then wasn't it? So what we want to do next now is trying to improve these source attribution studies. Are we getting a good representation of this local reservoir of infection? Can we sample other animals in the region? Can we sample the water? Can we build up a better collection for us to be able to understand what's going on in this setting? Are humans a source? Because we're now talking about asymptomatic carriage and chronic infection, is the same person infecting other members of the household, other members of their family? Can we look at co-infection with other Campy species or even the same species? Fran showed last week that in the chickens in the broiler houses, they maybe only had one Campy in their guts, but the healthy chickens were carrying lots of different Campy species, and maybe that's what's going on in this setting. And then something we actually did try to do with this collection, but it wasn't very successful because of the size of the data set, was to try and identify genes or SNPs that were associated with severe disease or asymptomatic carriage. Can we identify markers that will help predict if you're going to get very ill from an infection or not? So yes, this is just explaining some of the reasoning behind why we want to continue this frame of thought. So other sources of infection, what are we missing? So we do see, whenever we go out and try and sample Campylobacter, we can basically find it wherever we look. But often, they will be in sequence types that we don't see in disease. So in some of these papers, we see an ST in raccoons in Canada, but it's very different from the disease samples in Canada. Or there was a Helen Milner sampled rats in the UK, but again, it was a completely different ST to what we see in disease. And the wild bird isolates, every time we look in a different type of wild bird, we often find another ST that we see not very often in human disease. Or is there, or there's some lineages that are adapted to humans. So if we're thinking about human-to-human transmission within the same household or within the same family, are there parts of the tree or lineages that are adapted towards that? And there was one ST in this study where all of the ones we found were from asymptomatic infections. And when we go through the literature, we see hints that maybe this ST has shown this type of behavior before, where we see ST403 in the Dutch Antilles, and it lacks these genes that allow it to colonize in chickens, but we do find it in human disease. I think Francesca's mentioned ST2993 as well, which is currently part of an outbreak in Peru. So lots of people are picking up this one ST. It much more often than other STs, it can lead to Guillain-Barré syndrome. So it's quite a nasty ST. And usually we wouldn't see this very often at all, but we're seeing this quite a lot in Peru. And the only other times we've seen it is in these outbreaks of Guillain-Barré syndrome in China, South Africa, or in Pakistan. And yet all of this fits into what we're trying to do as a group, where we're trying to build up samples from around the world, from Africa, South America, and Asia, and look at different epidemiologies and see if we see this reflected in the genomes. And I think there's a cartoon. And one of the other things we do see as we go around the world is much more infection being attributed to these other Campy species, Campy coli, and other even less well-studied species. And if we look at coli from studies we've done in Egypt and Vietnam, we see a higher percentage of C. coli isolates, but in those isolates, they're much more able to pick up AMR genes and AMR elements. So that could be what Francesca has picked up in her paper, where she's collected over a longer period, seeing a lot of AMR, but she sees a lot more C. coli isolates in her collection. We didn't include the C. coli isolates in this paper, and that's a growing concern worldwide. And in one of the studies that we did, we were able to see that once the coli are able to pick up these AMR elements from different bacterial species and then spread it amongst themselves. So yes, we're hoping as a group to build this collection of non-European, non-American Campylobacter isolates and try and show people that Campylobacter is not always a food poisoning infection. There's also these other types of epidemiology, these other types of transmission events that can lead to a Campylobacter infection. Okay, well, thanks to everybody that is involved in all these studies around the world, loads of collaborators. But specifically for this work, I'd like to point out Margaret Kosek and her group. She's now at the University of Virginia in America, but also based in Iquitos in Peru. And Francesca, who works with Margaret as well, was highly involved in this work.